microorganisms that live. Oh, hang on. To describe the microorganisms that live in or a particular part of the body, such as the skin or gastrointestinal tract. So these groups of microorganisms are dynamic and change in response to a host environmental factors. So this is one of the phenomena, biological phenomena, that is nonlinear dynamic. In life system it's not linear. It's there is a dynamic. It's nonlinear dynamic. So and as a response to environmental factors such as exercise or diet, medications, and other exposure, and age included too. The more we age, or our age is also a part of the environmental factor, right, or exposure that would affect the composition of microbiome. So before we move on to the discussion, but at least I'd like to remind you there are three important jargons, like specific term that from time to time you will be exposed to, and this is need to know basis, right? So the first one would be the alpha diversity. So it describes the species diversity or species richness in an ecosystem. It is small scale measure. For example, if you want to figure out what is the gut microbiome of an individual, then the diversity or this richness will be reflected by the so-called alpha diversity. The second one would be the beta diversity. It describes the species diversity between two ecosystems or communities, and by itself inherently it is a large-scale measure. And the third one, gamma diversity. So it describes the species biodiversity in a large geographic region. So the data across the population in a particular large regions, and then you want to see how the diversity, how the biodiversity in that particular region, so would be determined by what we call the gamma diversity. So it is very large scale measure, definitely. So all these measures are important in protecting the biodiversity of the world. Now you would understand the interconnectedness of life due to the presence of microbiome, and you would get a good understanding too, based on the case of the COVID-19 viruses. And you can see just this one small virus, one small genome of virus, an RNA virus, not even a DNA virus. Then you could see the interconnectedness of human population due to the spread of this particular virus. But what we are experiencing in a daily life literally is not just one microorganism, but what we call a microbiome, an exposure to a bunch of like microbes. And this is enabled by expensive travel. So there is going to be a correlation. The more, let's say, in a country open up to travel, to not just tourism, but travel, the ability to travel across different regions would enable the dynamic, the exposure, and would alter or change the microbiome compositions in a population, subset of population. So now you get the understanding how serious it is, and the magnitudes, the scale of this importance, right? Now, on the other hand, this biases, also called dysbacteriosis, is characterized as a disruption to the microbiota homeostasis caused by an imbalance in the microflora. changes in their functional composition and metabolic activities, or a shift in their local distributions. So emerging evidence supports that rather than one or two dominant organisms, including the host cell. So the composition of the entire community of microbial residents influences a balanced immune response. Thus, the perturbations to the structure of complex commensal communities, or we call it the dysbiosis, the perturbation to the structure of complex commensal communities, or dysbiosis, can lead to deficient education of the host immune system and subsequent development of immune-mediated diseases. So this includes loss of beneficial microbial organisms, expansion of pathobionts or potentially harmful microorganisms, and loss of overall microbial diversity. So these three types of dysbiosis are not mutually exclusive and may all occur concurrently. So thus to re-establish a healthy complex microbiome after dysbiosis has occurred, a process we will refer to as a rebiosis will be fundamental to treating complex immune disease. So we got a snapshot yesterday about how to use metagenomic, but in the case of to develop a rebiosis by, let's say, fecal transplant. If you remember, 2 days ago, the lecture in 2 days ago. So in today's lecture, we will discuss the theory of developmental origin of health and diseases. So DOHAT, it's formerly Barker hypothesis, and we'll discuss cases of dysbiosis. So it's frozen. Anyway, yeah, so let's, and the next slides. According to the developmental origins theory or concept, environmental exposure during a crucial period of early life might alter fetal and newborn programming. So the Barker hypothesis originally, it's just, yeah. So I will, this is the previous one. So we got two objectives. So well, to explain the background here. So the microbiome has become increasingly important to human health during the last 10 years or so. And recent research has revealed that healthy immunological development and long-term health depend on the early life microbiome. So during this time, immune-mediated metabolic and neurodevelopmental problems have all been linked to transient microbial dysbiosis. The maternal and intrauterine microbiomes have also been shown to play a crucial impact in the development and health of children. So, and the research on this is getting more and more coming out, right? And these results taken together have been utilized to demonstrate the microbiome's role as a major contributor to the DOHAT. So the maturation of the early life microbiome and how momentary changes in this bacterial community microbiome can have long-term effects on human health. And thus become the topics of discussion in this lecture. Now, with regard to DOHAT itself, so the environmental exposure during a crucial period of early life might alter fetal and newborn programming. So the Barker hypothesis originally focusing on the link between fetal starvation, malnutrition, and hypertension in later life. Now, it has recently been broadened to include a wide range of early life exposure and birth outcomes related to long-term health and development. A high prenatal exposure to growth hormones, for example, may be the underlying mechanisms for the relationships between high birth weight and increased risk of breast and colon cancer, for instance, right? And although they were not initially linked to DOHA, early infections and microbial exposure were suggested as important environmental factors influencing baby immune development in the hygiene theory of allergy illness. And in light of recent developments in the study of the human microbiome, the hygiene hypothesis has been updated and is now known as the microflora hypothesis. So the hypothesis, the microflora hypothesis, postulates that early environmental exposure affect how the human microbiome develops. It is believed that changes in the microbiome's makeup can influence the immune system maturations to favor a hypersensitive and or hyperinflammatory state. For instance, the immune system's innate and adaptive components are both heavily involved in encouraging an inflammatory response. The T helper 1 response is generally elicited by microbe exposure. However, decreases the T helper 2 activity that is frequently linked to immune-mediated and hypersensitive reaction. And thus, an early crucial window of development for the microbiome has also been hypothesized. And similar to DOHA, this period's transient microbial dysbiosis, unhealthy microbial state, has been linked to long-term immunological and metabolic health problems, meriting investigation within the field of developmental origins. So that's how the theory or the DOHA has been developed. And in the image here, we see the timeline of microbiome maturation in one's lifespan. So from the early life critical window, comprising the conception, the birth, an infant, and to the early childhood, adolescence, adulthood, and finally the senescence. The infant microbiome is most vulnerable to the environmental influences in early life. So maternal to fetal microbial transfer, mode of birth, antibiotics, and diet can alter the colonization and maturation in the early life microbiome. These lifestyles induce variations in microbiome compositions and function, can have prolonged influences in human health and may lead to the development of disease later in life. Now we will discuss the early life critical window of the interplay between microbiome composition and the development of immune, metabolic, and hormonal systems at human life later on. Now, from conceptions to birth, it's an early development of microbiome within an individual. So the human microbiome is a composite creature. Organisms is made up of 10 trillion to 100 trillion microbial cells, including bacteria, archaea, and microbial eukaryotes. Viruses, including bacterial viruses or bacteriophage, and other microbes. That's within an individual human body. So compare this, the amount of cells in microbiome is surpassing the total cells that make up the human host, which is approximately 37 trillion cells. And in terms of the genomic catalog of this superorganisms, the microbiome, is made up of 3.3 million non-redundant genes. Imagine that, demonstrating the enormous functional potential of the microbiota. So compare this to the number of genes with compare this number with only 25,000 human host genes. So that means microbiome has way more capabilities to perform a variety of metabolisms than the host. And we know that we rely on gut microbiome to process food, even the strange one that we eat, right? Now, with regard to the phrases of microbiota and microbiome, this refer to the genetic database and microbial compositions, right? They are interchangeable in this field of study. So the following slides and discussion describe how human bacterial microbiome initially colonizes and establishes itself during infancy, from conception to the first year of life. Well, any questions so far? All right, so now from the conception of birth, so the first one, obviously, the maternal to fetal microbial transfer. Take a look here. Given that the fetus swallows amniotic fluid throughout the second and the third trimesters, the amniotic fluid and meconium is the first feces or stool of the newborn, right? So they compare the amniotic fluid and the meconium. May be expected to share a large portion of their microbiome, right? The microbiota. But indeed, yeah, studies have shown that the first feces of the newborn or meconium microbiome shares more features with amniotic fluid. So you can see an overlap here. And in this study, it shares more features with the amniotic fluid than with the maternal feces, placenta, colostrum, or infant feces. So there are 32 or 33% of detected species were shared between amniotic fluid and meconium samples in this case, in this study, while the 28% were found in the amniotic fluid only, and 39% were found in the meconium only. However, this 33% of shared species accounted for 94% of total reads. And so imagine that majority, it means majority are literally shared. Now, while the majority of reads recovered mapped to species that were shared between the two sample types, there were distinct differences in the relative abundances in these reads. So take a look here on the right side. So take a look, the meconium on the top one and the amniotic fluid on the bottom one of this bar. And majority of the family of the species belongs to Philomonas in here in the meconium. So indeed, you can see it in the graph here. The Philomonas purucae has higher count than Propionibacterium acnes. But on the other hand, the Propionibacterium here seems to have more abundance than the Philomonas. Now, interestingly, the Philomonas was strongly dominant in meconium samples. But was only present in 12 amniotic fluid samples and only dominant in 8 of That's from the data from the samples subjects. Now, importantly, subjects that lack Pelamonas in their meconium samples also lack it in their amniotic fluid samples. One may explain that this observation that Pelamonas may have a competitive advantage over other bacteria in the fetal gut. So meconium is very unusual substance made up of bile acids and salts, pancreatic secretion, epithelial cells, and the residues of swallowed amniotic fluid. And thus the Pelamonas, in this case, Pelamonas purucae, might be well suited to this environment, while other bacteria might be outcompeted or unable to survive in the meconium. Similarly, amniotic fluid samples were largely dominated by skin commensals, such as Propionibacterium acnes and Staphylococcus species. Right. Yeah. And which may be suited to such a needs, right? The amniotic fluids contain high levels of fetal skin cells, and these data may demonstrate bacterial niche development in utero. So the next one, the next exposure to the development of microbiome would be the birth mode, right? Here in this study, they compare they compare between vaginal birth versus C-section. So we call it veggie types of, and there are 90 women who delivered at term, meaning like longer than 39 weeks of gestation, and versus here on the right side, 45 women who delivered prematurely, less than 37 weeks of gestation, and showing 13 community states. So we call it the vegetatypes. And obviously, the way that the newborn was delivered, either through birth canal or C-section, both are the first step in the postnatal colonization. And exposure to maternal fecal and vaginal microbiotas would make up the would make up the microbiome of the newborn, the skin microbiome, and including the gut microbiomes. And we find after 24-hour, we've noticed the typical vaginal bacteria, Prevotella and Topobium species were also detected in the neonates. You can clearly appreciate. So, and with the term birth, this majority here, the cluster of Streptococcus, and there is a Lactobacillus. Different patients would have differences. So there are patients that would be pretty much detected, would be Streptococcus, let's say cluster 29 here, versus there is a subject with predominantly Lactobacillus crispatus. Or some of the patients would have quite a variety of detective of microbiotas, right? Any questions so far? No. In this study, you will see in the number of shared and distinct OTUs. 2 days ago, we talked about OTUs and the operational taxonomic units between vaginal here, here blue, and 1 week fecal, the microbiome that has been collected from this sites, and that's just the 1 week fecal, the purple one, and the airways in here, right? And with the total, this is the total 2,000 something, 2,300 OTUs were detected. So the percentages indicate the relative numbers of breeds of the artisans. So there are 319 ubiquitous OTUs account for a total 88% of the reads. And the ages between OTUs indicate the pairwise correlation. So reflecting the co-occurrence based on correlation network analysis on each compartment separately. So using the highest value among the 3 compartments. Now the thickness of the edges, here, this is the correlation. This is just an illustration how intercorrelated, how it would be an overlap. So let's say between the airway might not be, but there is an airway and vaginal, and then this fecal and vaginal, and it has been plotted to give you an illustration. This is an illustration to provide you with the richness of the OTUs. In this case, it's OTUs is the unit, and you can see here the variety of microorganisms detected, and also on here on the bar graph here, the microbial composition in pregnancy, at birth and after births, and then you can see, you can appreciate here. This is the week 24, week 36, and the birth, and then the sample from the feces and the airway, and then so you can see that there is a dynamic changes during the colonizations, the pre and post natal colonization of the microbiome. So the microbiome development and the changes occurring, and this is what happens. And on top of this, later we discuss this biases, and that what happens if this being perturbed and this biases occurs, and it might affect the long-term health of the individual. Now, further investigations, and we can to compare between vaginal birth and the C-section. Here, the end from 64 subjects, and the C-section of the 68 subjects, and you can clearly see which of the families would be missing during the C-section. So now you can clearly see it's quite significant. So if we expect that the normal delivery would expect the diversity, however, the intervention of the delivery by cesarean section would alter and would eliminate a variety of them. Yeah. Now, to summarize, the differences between this normal versus the C-sections. So we have 4 columns here. What is the observed association with cesarean section, and what is the microbial group affected at the genus level, and what is the specific disease health condition that would that were found to be correlated with, and these are the references. So first, the decrease or delayed colonization after C-sections, and its effect Bifidobacterium, Lactobacillus, Bacteroides prevotella, and the imbalances of bifidobacterium or the missing bifidobacterium apparently has been linked, at least statistically, to necrotizing enterocolitis, eczema, obesity, liver disease, colorectal cancer. And how about lactobacillus? It links to atopy or obesity. And Bacteroides apparently would link to type 1 diabetes, and Prevotella also would link to some inflammation in type 1 diabetes. So how about increased colonization after C-section? If there is an increase of Staphylococcus, then there is a atopic dermatitis associated health issues that has been observed in the populations. If it is Clostridium, again, atopic diseases, including dermatitis and asthma. So now you can see there is a link, and we cannot just dismiss. Okay, the C-section, mostly in numbers of countries, would be the option, the choice of a new mom to deliver the baby, but there is a health implication that we have to consider. Okay, and the next part would be also the breastfeeding. So breastfeeding also critical, important to build the microbiome compositions of the infant. So the potential of breast milk to prevent allergic disease by shaping of the neonatal gut microbiota. Here it's illustrated here in the heat map. The breast milk, as we know, contains microbes as well as factors that indirectly shape the gut microbiota of the neonate. Breast milk can direct the early microbiota composition. For example, the favor the growth of bifidobacteria and lactobacillus, and affect microbiota metabolic function, which subsequently can impact on immune development and maturation. The gut microbiota in early life impacts on immune maturation via microorganisms associated molecular patterns signaling. So for example, via microbiota metabolites such as short fatty acids or aryl hydrocarbon receptors ligands. So the gut microbiota of the neonates can direct the immune system toward allergy prevention, for example, via the induction of regulatory T cells direct, which contribute to oral tolerance induction, and interleukin, so such as interleukin-22 production by innate lymphoid cells, for example, which strengthen the gut barriers. Now, as a result, shaping the infant's gut microbiota by breast milk has the potential to direct immune system toward allergy prevention. So, and in this study, from the homogeneous microbiome would diverges in the gut microbiota. So, and becomes microbe-specific body niches. And so here we focus on the gut microbiome, the data, and it's maturing up to 3 years old. And of course, the breastfeeding, the breast milk would, we know that the composition would depend on the nutrition and the mom nutritional status as well. But the infant diet, mom would provide the infant diet, an additional infant diet, or would be differ than other moms, for example. And we are talking about breastfeeding, and of course, if you use a bottle feed with not breast milk, that you can imagine it would alter the compositions of the microbiome as well. So here is the study of 107 mother and infant, and it was breastfed for 30 to 40 days, and 28% from breast milk, the microbiome, and temperature from areole skin, and we can and the dose dependent, meaning the composition and proportion of daily breastfeeding also was contributed, was calculated, and as I said before, the formula fat apparently decreased diversity. So and then now you can see it's on 40-day, this is 3 months, 6 months, and so they compare breastfeed group and the formula feed group, formula B feed group. So so the 3 boxes, the 3 columns here from the breastfeed group versus formula A feed group and formula B, and then we've got to the conclusion is that there is a decreased diversity, and you can see let's say we started with 40 days here, and then you can see in 6 months, and then you compare the 6 months, and it's definitely in the calculation you can see there is a differences in the diversity. Well, this is not quite clear in the first instance how the quantifications would tell you, would show the reduction of diversity in the formula fat group. So, but the take-home message is that the breastfeeding is critical to the colonization, to the developmental maturations of the newborns or the infants' microbiome, in gut microbiome. And here is a little bit more toward the mechanisms. I think I explained this. The mechanisms. Yeah, I forgot this slide. So I was explaining the slides, but this is how the microbiota and the breastfeed would affect the microbiota, and how the microbial proliferations to human males, and also there's an antimicrobials that would prevent infections, and this is the explanation that I mentioned to you, for example, to short chain fatty acids or hydrocarbon receptors. So this is gut epithelium, and this is the interplay between the bacteria, the gut microbiome, in this case lactobacillus and bifidobacteria, and through HR ligands or FA production, and it would trigger the innate immune system's signaling and would prevent the allergy. So that's all right. So now what happens if there is an imbalance during the critical window of early life? So this is the term invent. So if you check the intestinal microbiota, what happens to the dysbiosis of this gut microbiota? So there is a home environment would affect, and the feeding type, and as we we should go from the top, from the hospital environment, right? The hospital environment, and in the hospital environment, and then maternal microbiota would affect the gut microbiomes, and the mode of delivery, we discussed either through normal birth canal or C-section, and the feeding type. So we discussed about the formula fed or breastfeeding, and obviously the home environment. So and during the antibiotic exposure is also affecting the intestinal microbiota. So, for example, if the baby needs to be um, treated by antibiotics, and the feeding tube uh, allow the formation of biofilm, and that obviously would affect also the balances of the microbiome. And so we'll discuss this one, and then we'll discuss the right part to affect the necrotizing enterocolitis, late onset sepsis, neurodevelopmental impairment, colic, atopic diseases, and diabetes type 1. So as I mentioned previously, so dysbiosis, in short, unhealthy microbial state. So a narrow definition of dysbiosis is a stable microbial community state that functionally contribute to the etiology, diagnosis, or treatment of a disease. And dysbiosis is often driven by infection and inflammation, diet, and xenobiotics, host genetics, or the host environment. It is a complex system that would, so it's not by one particular aspect. It's not necessarily, but there are an array of exposure that would lead to dysbiosis. Innate and adaptive immunity control the colonization of needs of the intestinal microbiota through mechanisms including the production of antimicrobial peptides and IgA antibodies. So the interplay between the IgA antibodies in the mucus, for example, and the antimicrobial peptides that would kill off or would suppress, would prevent a proliferation of certain species of bacteria. And a dysbiotic microbiota may actively influence its colonization needs by altering the functions of innate and adaptive intestinal immunity. And so we know that dysbiosis has been associated with many immune-related human diseases, but in many cases, it remains to be established whether dysbiosis is a cause or consequence of the disease. So this is a critical thought to figure out, to find the point whether it's a cause. Actually, the dysbiosis, when the dysbiosis happen, then it's the long-term effects such as obesity or type 1 diabetes, or actually the consequence of the disease. If the infant become sick, and it will lead to dysbiosis. So that's if the dysbiosis become the consequence of a disease, and also it will affect later stages, the health status of the individual at later stages. And personalized nutrition and metabolite-based postbiotic therapy might present ways to harness the increasing knowledge about dysbiosis in disease or the design of new therapies. So this is the promise why studying dysbiosis, and so and this is linked to the nutrigenomics. Nutrigenomics, we want to understand the genomic aspects of the nutrient, the diets, and especially in the early infant, early life, it's quite critical. And the diets, the nutrient here, if we want to study, if we use genomics to study, and so we want to figure out what happens in the microbiome level, at the microbiome level. And so the study, for example, there is a study in Japanese macaques. It's a long-tail monkey macaque study with a high-fat diet induced dysbiosis in the offspring microbiome. So now you can see the nutrient component could induce the dysbiosis. So imbalance or unsuitable diet might induce dysbiosis in the offspring microbiome. And low-fat control diet post-weaning apparently failed to correct the dysbiosis. Now, there is a human prospective study to understand the gestation or breastfeeding associated maternal diet. So high-fat maternal gestational diet induces variation of meconium microbiome and persisted to 4 to 6 weeks of age. It's still plenty of questions from these two studies. They're quite unanswered questions because of the complex ecosystem. How about pre and postnatal antibiotic exposure? Pre meaning during the, for example, if the mom got sick and needs antibiotic, if there is an infection, and vice versa, after the birth of the newborn and the exposure to antibiotic, definitely for both situations would be an early major stressor on the infant microbiome. It would alter diversity of both maternal and neonatal microbiome. So now you imagine a pregnant mom would, if a pregnant mom would take an antibiotics, and you can now you can imagine that both, not just mom, but the fetus also have got affected because of the antibiotic exposure. So the intrapartum antibiotic exposure also alters infant oral microbiome. So the study found there is a decrease in Streptococcus, Camelacea, and Lactobacillus, but enriching proteobacteria. And of course, it depends on the antibiotics types. The antibiotic-induced dysbiosis in infancy would lead to various non-communicable diseases in child and adulthood, such as obesity, asthma, and so this is the issues, the IBD, so inflammatory bowel diseases. And so the early life antibiotic and versus immune deficiency or affected immune system. So this interplay still need to be elucidated. Now, how about non-communicable diseases? On the panel, the right panel, and the first that is going to be affected, for example, necrotizing enterocolitis, preterm infants. So this is a neonatal microbial dysbiosis. There is a correlation between NEC and then the neonatal microbial dysbiosis that happens in preterm infants, and it can be modified by antibiotic exposure. So the onset of NEC has been shown because of the abundance of Proteobacteria, Firmicutes, and Bacteroidetes. And the similar microbiome variation in low birth weight neonates also has shown the abundance of Proteobacteria and so on. Now, the increase in Proteobacteria versus the increase of enterocyte, toll-like receptor 4 activity. So the microbiome versus the immune system, the immunosome. So that would be inducing a hyperinflammatory response to a dysbiotic microbiome. And for example, a significant risk, an easy marker is an increase of uropathogenic E. coli. And there is a study when the treatment of the antibiotic between 7 to 14 versus 0 to 6 days would lead an enrichment of the E. coli. How about the prevention or the treatment of the NEC, and whether probiotic supplementation of preterm neonates would help? And is there any probiotic strains, right? Now, I remember something. There are some practices by gynecologists after the doctor would deliver the baby. through C-section, and the doctor would sample vaginal fluid and rub it into the body of the baby, so that to offset some of the dysbiases or imbalances that it's going to be missing. So during the C-section. Now, asthma and atopic diseases, AAD, it's been known becoming an issue and becoming more related, and some link has been shown due to the dynamics of gut microbiomes. Although there is still two sides of the coin, whether it's microbiome or developing immune system in the neonates would play a role. The food allergies and dermatitis has been shown a strong influence by commensal microbiome. So, and the mouse model of allergy airway inflammation shows this evidence of such association. And postnatal early life dysbiosis in both gut and airway microbiome would link to atopic diseases and asthma in children. And it is important, although this is transient, but it's prominent between birth and the first 3 months in the infant. So the dysbiosis occur in this period, and it would affect and would link to this AAD. And so another issue is food sensitization among 1-year-old children due to the decrease of microbial diversity at 3 months of age, and increase of Enterobacteriaceae or Bacteroideaceae ratio at 3 months and 1 year of age. And well, the developmental origins window for AAD apparently from birth to 1 year of age. That's a critical part that would determine whether at later stages would link to the AAD. The next one, obesity. And the temporal or compositional microbiome versus early childhood adiposity. And this is also critical. And some study has found the link between this obesity and the early childhood adiposity, meaning the development of adiposity during the early childhood. And so it is critical, and we found, and from the study, there is a high abundance of Streptococcus species at 6 months of age, and the increase of adiposity at 18 months of age. So it's seemingly unrelated. We might undermine just for example here in this case Streptococcus species. At 6 months, apparently, there is a statistical significance of adiposity way later in the infant's age from longitudinal studies. And apparently, the mouse model of such studies would also important to understand the link between the obesity and the dysbiosis of the microbiome. Now, the next one would be the neurodevelopmental disorder. And there is a theory about gut-brain axis. So, and it becomes quite a topics of discussion, whether microbiome compositional variation would correlate with how the people cope with stressful situations and how they respond to stress and the anxiety that could happen. Now, in the mouse model studies, the neonatal stress after maternal separation would apparently impact the long-term microbiome changes. And how about a probiotic treatment? It might counter the elevated cortisol level. So, however, this is still not solving the issues whether it has a direct correlation, but the indication is that that could be a role to play among these two parameters. And there is a study that to figure out that SCFA, propionic acids, apparently increase anxiety-like behavior if the exposure occurred in before the birth of the newborn, and such prenatal stress obviously would alter placental microbiome. How about impaired social behavior, like similar to those suffer from autism or creating an autism spectrum disorder? There is a study to link the reduction of Lactobacillus reuteri to development, the neurodevelopmental disorders at later stages, but it is still going to be difficult to pin down mechanistically. Well, others such as type 1 diabetes, this is really critical because it's chronic non-communicable disease. Whether early life microbiome composition would lead to type 1 diabetes. And how about the link to cancer, such as breast cancer? And what is the microbiome DOHAT role? And so the study of microbiome, the metagenomic study, to understand and perhaps the multi-omics study would now open up an array of other potential interplay and by itself numbers of questions. And so, for example, in the future direction here, the pre and post-birth exposure alter the microbiome compositions and functional potential in the neonates. So it still has to be pinned down, pinpoint, and a different population, demographic, populational demography, and how microbiome plays causal role during critical window of developmental plasticity. This is even more challenging to figure out, and as I mentioned, the multi-omics or combined omics analysis, multi-omic analysis might provide a better understanding of microbiome role. So because we would get some here and there some link, for example, the enzymatic differences in the proteomic study that would alter the SCFA, for example, and how the genetic expression would correlate with that compositions. Well, we are talking in a biome level, and how the scientific methodology, the development, the improvement, the advances of in the scientific technology will help to provide a better understanding the interplay, the interaction of in life and in the biome. So, any questions so far? So I'm gonna stop sharing, and then we can have a discussion and question and answer. I think it's still pretty quick. Still. Okay.
Not a question, actually, just a reflection after listening to your lecture, Doctor, because my role is in the ministry is more on the program implementation. It is quite useful for us for the program implemented, because for the nutrition program, when we provide the nutrition education, we promote the exclusive breastfeeding, and the more we can highlight to the community is more about the nutritional benefit. But now I have learned that the effect, the benefit of breastfeeding on the gut microbiome composition on the infant and its later life risks for, especially for my interest
Mhm.
for the chronic non-communicable diseases is really useful because
Mhm.
I may not have in-depth knowledge about the detailed methodology, but finding that those kind of microbiome composition and also the later life chronic diseases, non-communicable diseases is really useful for us for the program implementer that we can provide more information with more confidence to the community. Please
Mhm.
give breast milk to your baby, something like that. Just to share the
Right.
reflection, doctor. Thank you.
Yeah, that's awesome. That's wonderful. And we've got more insights by this microbiome metagenomic studies and an exclusive breastfeeding up to 2 years if possible. Some moms would feed all the way to the third years. And on the other hand, and how to mitigate the lack of uh, the breastfeeding. So um, that's something that is critical during the policy development, and I totally agree with you. So this will give insight, will give more uh, reasons to prevent uh, well, in the population study, this is one of the benefit for public health. So in the public healthcare system, such epidemiological study is really important. Why it would we would try to overcome issues later on. Then issues and health issues would correlate with high economy costs, right? So uh, I think it's yeah, it's wise and it's important, and take the advantages of this study and then persuade the stakeholder, the government, to invest more on this kind of study. And as a leverage, it would help to overcome the current issues with all you name it, all the diabetes, the issues with diabetes, the obesity, the neurodevelopmental issues, and at the same time prevent the in the next generation help would help to prevent such from happening. And but at the end, the importance of the data is to harvest it, to understand it. What would be the local population? What would be the pictures of the microbiome in the local populations? And anyway, others might want to have a comments or discussion or questions, and so please feel free.
Dr. Felix, excuse me,
Yeah.
I would like to ask you about the question. So we live in Lombok, West Nusa Tenggara in Indonesia. And in here, in our district, there are a lot of anemic children and also stunted children. So in your experience, is there any different about the microbiota between the anemic and non-anemic children, or also in stunted children compared to non-stunted children about the
Right. Yeah. Good questions. And the first instances, once my suggest there's going to be differences in the microbiome compositions between the anemic, the stunted infants or children versus the normal rates, right? And all in all, depends on first the diet. The diet is critical. So and I I'd like to take the examples of there is a genetic factors for sure. Let's separate these genetic factors, the environmental factors. So one of the tallest how do you say, nations, people in one of the tallest nation would be the people from the Netherlands. And apparently back several hundred years ago, they got used to, you know, like a milk and drink milk. So, and until today, they got used to drink milk. And apparently over time, the generation after generation, there is an increase in height and weight. So, and we know the nutrition critical to prevent the issues of malnutrition, or even the phenotypic you know, like implications such as stunting and so it's replicated in different nations. Those people of Japanese and Korean, the next generation is getting less stunting issues. And now you might wonder if not just purely the diet as a factor. So let's say from the policy making perspective, how to provide nutritious component element accessible to any children, that undergo rapidly development from the neonates all the way to for sure like at the elementary or primary education. So, and yeah, that's you're going to be wondering what is the dynamic in the microbiome system. So this is what the importance of the epidemiological study. You have the range of a normal set of the normal development, and if you were to collect the microbial data or microbiome data, and then versus the other group that has an issue with anemia, and the other group that has an issue with the malnutrition or stunting, for example, then you would be able to pin down on which one of it, and you might be able also to correlate if potential probiotic treatment would be one of the potential answer. That's from the point of view of nutrigenomic alone. But we in nutrigenomic, if we were to step up to have a multi-omic approaches, then you can such, I think to Mifa mentioned or Dr. Lane mentioned I think a couple of days ago, mentioned about the study of using epigenomics, for example, or at least at the DNA level. We want to figure out if there is a genetic factors that has been an inheritance issues that might lead to, in your case, probably stunting or anemia, for example. That has to be addressed. So that's the benefit of the multi-omic studies. Yeah, having said that, as far as if there is other study that would describe, just a matter of searching the literature. But in the first instance, the answer to your question, yes, there would be a difference, and that's something it's in need in the epidemiological program, or in for Department of Health, for a populational health. Right.
Thank you, Dr. Felix.
You're welcome. Dr. Lang asks the question, home environment as one of the determinants of infants' intestinal microbiomes. Can you please a little bit borrowed some examples of what does it mean, the home environmental. Yes, this is really good question. Why home environment would we would have to consider it as one of the factors here in this case that would determine the intestinal microbiome. So I know this is kind of a stretch, but and it is still important to include this category as especially in the developing country or underdeveloped country. Then you would imagine urban, rural, suburb, even suburb would have play a critical role. And the rural area, such as in agricultural setting or in the fishery settings, some countries, I think, including Indonesia, there are still numbers of population who lives on top of seawater, for example. And this is going to be a totally different exposure than those in the urban area or dwells in high rises in the metropolitan city. So this is important also to the development of. I know it's hard to correlate right away, but the ability to there is a tug of war or an arm race between the immune system development and microbiome compositions, right? So, and the immune system development would curb the microbiomes, and vice versa. The microbiome dynamic would trigger the development, the maturation of the immune system, and maybe we could, other than diets, I know the diets might be different, those in the rural area versus those in the urban area, those in the modern environment that mostly eat like microwave food, frozen food, versus those who just need to catch the fish or and then process it right away. And so if we eliminate these factors, but the immune system development itself would directly affect, like I mentioned earlier, there is an intimate interplay between this microbiome dynamics and the immune system. Otherwise, if the immune system has been weakened by particular exposure, particularly a disease, like if in the rural area and this infant would be exposed, let's say, unfortunately got a dengue fever, and that's weakened the numbers of immune system, and then would undergo dysbiosis, and numbers of now a pathobion, like potentially harmful microbes, is proliferating faster. So that's going to be the imbalances that happens. So I hope this could provide you a bit of a picture to why the home environment it's critical, and you can even go into the detail into one particular environment. So within the same urban area, within the same, let's say, an apartment in a skyscraper, but one has extremely sanitary versus the other one would be couldn't care less with all the environment, with all the organic matters, and then would lead to, let's say, attracting more flies. And yes, that's gonna be creating a difference, Steve. So I hope it will give you a perspective.
Yes. Thank you for detailed explanation.
You're welcome. And Ibu Ami, the gut-brain axis, the question about the gut-brain axis. Is there correlation newborn babies, some stress or depression mother in microbial babies? That's really good questions. so far we've got all the connection. I mentioned when the mom got sick, needs an antibiotics, and obviously it would affect the baby too. And it would affect all the way until the baby was born, for example. And the stress and depression would definitely would alter compositions of microbiomes. It's simply because the not just the hormonal, the chemicals, the secondary metabolites change. You know, when what we are talking here, stress or depression is a prolonged uh, prolonged stress. So depression means a prolonged stress. It's not just like 1 hour you've got depressed with such a gloomy news or for sudden sad news, and then but the next day you recover. And no, how about a prolonged stress or depressions? And we know that a depression would suppress the newborn development in not newborn the fetal development in the womb. So, and it would affect. We were just talking about stunting, and consider this. Consider that prolonged stress or a depressed mom would also affect the growth, and it might lead to stunting. So literally, this is not a genetic inherited factors, but environment factor, because the fetus the fetus is still dependent on the mom's well-being, right? And I understand that this is affect many women, and I understand that there are period of prolonged stress in mom in a pregnant mom, so in pregnancy. So in the more we have better off in the exposure about this situation, the better of the social support being established, being built. So the only way is not like not just biological or medicine. Medicine is one thing, but you wouldn't want to expose a pregnant mom with different kind of antidepressants, right? So I've been at one point licensed to by the Health Canada in here to send off controlled substances, and it's incredible that people are in pain, people are depressed, and so many people would take the antidepressants. But if you're a pregnant mom, the healthier way, the first approach would be the social support. The social support, and this is at the level of not just government, but might start both ways: the top, the level of government, or the bottom, from the bottom, the level of family support, the security feeling, and to recognize what factors that create the anxiety, the prolonged stress. And so yes, that's the answer. But the gut-brain axis within one individual, that's also something. So the gut-brain axis in the mom, yes, it's a fact, but not just that, the psychological situation or the brain axis of the mom would affect the newborn of microbiota. So I hope it would answer or at least provide you some perspective, Buemi. Any further questions or discussions? Anyone else to have questions? Okay, yeah. So, okay, I think today we cover some of the microbiome. So next Tuesday, we're supposed to talk about human microbiome and then talk about this contributions to diseases but because next Tuesday, I'm supposed to also to showcase a data and how to analyze. So I think it's good to have an introduction about microbiome. At least you've been exposed to human microbiome, got some understanding. So maybe we can just expand a little bit, but a little bit more toward the basics. But importantly, we'll see if I can just to showcase how we have a data, metagenomic microbiome data, and how to analyze. Although this is not a computer sciences, but hopefully you've got some like a perspective to fill the gap if you have a data. I mean, you've learned how to run a study a couple of days ago and how to think about a study and then how to use the tools, the technology in the omics, including metagenomics. And now we've discussed the contributions of such study and how to decipher a complexity in the it is still in the field of nutrigenomics because it correlates with the diets, the nutrition, the environmental factors, and the microbiota literally survive because of the diet. So and although we have a uniqueness, each of us would have a differences in our diet regimens and would translate into the dynamics of differences in microbiomes, but we could find a pattern in a population, subpopulation, and whatever the status, including the questions with regard to the stunting, for example, and next we will see how we manage the data, and the principle will be given, but at least at one point you are as a principal investigator, you've got an overview, an overall pictures, how to design a study, how to check or ask questions, how to develop a methodology, and what will be the contributions to the society. And so the missing point would be, what does the data look like, and how we can analyze the data. And anyway, so if there is no more questions, so I hope to see you all again next Tuesday, and I wish you all the best time and the rest of the week. Enjoy the rest of the week. Thank you, everyone.
Thank you very much.
Thank you.
Yeah.
Doctor.
Bye.